Hello, everyone. Welcome back to my video tutorials for Pesudo-bulk RNA sequencing data analysis. In this video, I'm going to show you more complex edgeR pipeline, and I will demonstrate how to use edgeR to identify mark genes for different cell clusters from single-cell RNA sequencing data analysis. In video 1, we integrated 3 healthy lung dataset with 3 IPF lung dataset, and we created a threat object named as the control IPF combined RDS. So in today's video tutorial, I'm going to use this threat object for demonstration. If you want to use this threat object for practice, you can watch my first video tutorial for Pesudo-bulk RNA sequencing data analysis. So now we need the library thread, lemma, and edgeR. Then we can use the read RDS function to load the data. Let's load the thread object in. You can see we loaded the thread object. We can have a look at the metadata for the thread object. You can see the row names and the barcodes. We have the sample types in the origin ident, and we have the cell types here. In video 1, we annotated 5 cell clusters. We can have a look at the cell types. Let's run. You can see we have the fibroblast, immune cells, endothelial cells, epithelial cells, and the parasite smooth muscle cells clusters. We can use the table function to have a look how many cells in each cell cluster. Let's run it again. You can see here is the cell numbers for each cell cluster. We can look at more information for this thread object. We can use the dim plot to look at the cell clusters. We can plot the samples in the origin ident as plot 1, and also plot the cell clusters as plot 2. Now we can visualize both plot together. You can see here is the UMAP plot. Let's zoom in. So you can see on the left-hand side, the cells were colored by their samples names. You can see we have 6 samples, control 1, 2, 3, IPF 1, 2, 3. From the color, you can see the cells were well integrated into the same cell cluster from 6 different samples. And on the right-hand side are the cell types. You can see immune cells are in red, endothelial cells are in green, parasite and smooth muscle cells are in purple, fibroblast in blue, and epithelial cells are in dark green. So we know the cell type information from the thread object. Because we performed the data integration, now we can set the default cell as RNA. So next we can create Pesudo-bulk samples from the thread object. We are going to use the thread to Pesudo-bulk function again from the edgeR package. So the thread object will be control IPF combined. For the sample argument, we know the samples are in the origin ident column. For the cell cluster argument, we know we have 5 cell types in the cell type column. So now we can create the Pesudo-bulk samples. Let's run. You can see we generated a large DGE list. We can click the DGE list and have a look. You can see we have 3 elements, the count, samples, and the gene information. You can see in the list we have 20,680 genes. and 30 samples. So you can also use the dim function to look at the gene numbers and the samples numbers. We can run. You can see we have 20,680 genes, 30 samples. And also you can get the count data from the DGE list. We can run. If we click the count, you can see the row names are the genes, and the column names are the cell clusters for each sample. Because we have 5 cell clusters for each sample, and we have 6 samples, so in total we have 30 columns here. And also we can get the samples name from the DG list. We can run it again. Let's click the samples. You can see here we have 30 samples, and the first column is the group. But at the moment, all the groups were labeled as 1. Later we will change the group into control and IPF. The second column is the library size. Then the third column is the normalization factors. And also you can see at the moment the normalization factors for each dataset is 1. Later when we perform library size normalization, you will see the changes for the normalization factors. So this column is the sample information and the last column is the cell clusters information. So now we can change the group name. We know the first 15 samples are from control samples, and from 16 to 30 they are IPF samples. So we can set the group as the control and the IPF. Let's set the group names. Now we can give the group names to the sample names in the DG list, and we can get the samples again. If we click the samples, you can see now we change the group name to control for the first 15 samples, and then from 16 to 30, the group name is IPF. So we change the group name. Next, we can filter the data and also normalize the data. So I showed you the library size in the samples. You can also use the summary to look at the library size in the samples. We can run. You can see the minimum library size is nearly 140,000. So normally if we filter the library size, we filter out the library size below 50,000 because all the library sites are bigger than 50,000. So we actually don't need to filter the library size. But I still run this code in case your samples has library size smaller than 50,000. You can run this code. Let's set the threshold as 50,000. Then we can have a look at the keep samples. You can see because all the library size in this thread object is above 50,000. So all samples were labeled as true. And if we use the table to have a look at the keep samples, you can see 30 samples are true. Then we still perform the filtering, but we actually didn't filter out any samples. Next, we can filter out newly expressed genes. We can keep genes for the minimum count above 10 and the minimum total count above 20. So let's use the filter by expression function to filter out the genes. If we use the table function to look at the keep genes now, you can see we have 8,613 genes labeled as false, and 12,067 genes were labeled as true. So if we look at the DGE list at the moment, you can see it is still 20,680 genes. If we filter out the lowly expressed genes, let's filter it, and we can have a look at the DG list again. You can see now we only kept 12,067 genes that have high expression. So next we can normalize the library size. So for the library size normalization, we perform TMM normalization to estimate the effective library size. So let's normalize the library size now. And we can get the samples from the DG list again. Now let's have a look at the samples for the normalization factors. You can see the normalization factors will change for each sample. You can see the small library size has the bigger normalization factors, and the larger sample size has the smaller normalization factors. So after the library size normalization, we can have a look at the data. First, we can get the cluster information from the DG list. Then we can use the multi-dimensional scaling plot to look at the samples. We can run. You can see we generated the plot. Let's add the cell cluster information into the plot. Now we can zoom in to see the MDS plot. You can see here is the plot. We have 4 cell clusters here. You can see samples for endothelial cells were labeled in black color. Then epithelial cells were labeled in red. Immune cells were labeled in blue. Fibroblast and parasite smooth muscle cells were labeled in green and light blue. Because the fibroblast, parasite, and the smooth muscle cells are mesenchymal cells, so they have been clustered into one cluster. So from the cell cluster, you can see the bulk RNA sequencing data analysis produced a very good result because samples from the same cell type were clustered together. So now we can continue our analysis to identify marker genes for each cell cluster. So first we need to design a matrix. So this is the more complex edgeR pipeline. You know, in the thread object, we have 2 types donors, control and IPF. So we can get the donor information from the group column. Let's run. And also we have 5 cell clusters in the thread object. Because we want to identify differentially expressed gene, there will be marked genes for each cell cluster, so we can use the model matrix function to design a matrix. So here will be the cluster. We can also include the donor information, so we can design the matrix. So if we designed the model matrix to include the cell cluster information and also the donor information, so let's run. Then we can have a look at the design. We can click it. You can see we have 30 rows here because we have 30 samples. And we have 6 columns here. You know, we have 5 cell clusters in the data set. But here, the first column, instead of using a cluster endothelial cells, the design matrix produced an intercept column. So the design is correct because I just measured the baseline of log count per million expression for the endothelial cells cluster, then compare with other cell clusters. Then the last column is the donor information. You can see for the control it is 0, for all the IPF they are 1. So in this design, we included the information for both clusters and the donor information. But for today's analysis, we only want to identify mark genes for 5 different cell clusters. We don't want to identify the gene difference between control and the IPF, so we don't have to include the donor information. We can just design a model matrix for the cell clusters. Let's run it again. You can see now for the design matrix, we don't have a column for the donor information here. So we are going to use this matrix and perform the rest analysis to identify mark genes for each cell cluster. So we designed a matrix for the cell cluster. Now we can use the design matrix to estimate the dispersion for the DGE list. So we can use the estimate dispersion function to estimate the dispersions. Let's run. So this function will estimate common, trended, and tag-wise dispersion. So after this, we can visualize the dispersion using the plot biological coefficient of variance. So let's plot the BCV, and we can zoom in. You can see the tag-wise dispersion are in black dot, and the common dispersion is the straight red line because the value is the same for all the genes. Then the blue line is the trended dispersion. So next we can estimate the quasi likelihood dispersion using the generalized linear models. First, let's estimate the quasi likelihood dispersion. We can run. Then we can visualize the dispersion with the function plot quasi likelihood dispersion. Let's make the plot. We can zoom in again. You can see the red color squares the dispersion, the blue color shows the trended dispersion. So now the data is ready to identify mark genes. First, let's get the cluster information. We can set the cluster as n levels. Then we can make a contrast for each cell cluster. Later we can use the contrast to identify the mark genes. So we can have a look at the contrast. You can see at the moment we have 5 rows and 5 columns. And all the data in here is minus 0.25. Next, we can change the value in the diagonal box from 0.25 to 1. Let's change it to 1. You can see now the values in the diagonal box are 1. And then we can change the first row as 0. We can run. You can see now the value in the first row is 0. So the values are ready. Now we need to set the column names and the row names. We can get the row names from the design matrix. You can see now the row names are the intercept, cluster, IP, fibroblast, immune cells, cluster, parasite, and smooth muscle cells. And also we need to set the column names. We can get the column names from the cell cluster's name. Let's run. You can see now the column names changed to the cluster name for each cell type. So we make the contrast ready. Now we can create a QF list. So now we can perform the GLM-QLF test to identify the mark genes for each cell cluster against the rest cell cluster. You can see the comparison is for the cluster against the other cell clusters. Let's run. After finish the F testing, we can have a look at the KLF data list. You can see here we have 5 data lists. At the moment, it labeled as 1 to 5. We know the data in the first one is for the endothelial cells cluster. The second one is the epithelial cell cluster. We can click and have a look at the data inside. You can see that's all the information in the list 1. The most important part is the table. You can see in the table we have the log FC changes for each gene. We have the log count per million, and also we have the p-values for each gene. So we can get the table for the endothelial cells cluster. Let's run. We can also get the table value for the epithelial cells. I just use endothelial cells and epithelial cells as examples. You actually can get the table value for all the cell clusters. So let's connect the endothelial cells and also the epithelial cells to have a look at the genes. First, we can connect the endothelial cells. You can see we have the log FC value, log CPM value, the F value because we perform the F test and the P value for each gene. And here it includes all the genes, 12,067 genes. If we reward the log FC value, and we can have a look now for the endothelial cells, you can see the genes are ordered by highest log FC value to the lowest log FC value. We can have a look at the genes here. You can see the EMCN and SOX17, SOX18, and also TIE1. So we know they are marked genes for endothelial cells. And also you can see CLDN5, we always use this gene as a mark gene to identify endothelial cells in the lung. So next we can connect the epithelial cells dataset. And we can reward the log FC value again. So you can see now from the highest to the lowest, we can see the SCGB3A1, KRT15, and also we scroll further down, you can see KRT17. Those genes are the mark genes for the cluster cells. If you scroll further down, you can see SCGB1A1. And also going a little bit further, you can see KRT19, and also here is the EPCAM. This is the mark gene that we used to label all the epithelial cells. So here, because we included both data from healthy donors and IPF donors, so some genes presented here are not classic mark genes for the cell types we identified for healthy donors. So I used the endothelial cells and the epithelial cells as examples to see the table for the log FC changes p-values. If you like, you can also look at other cell types. Now we can use the LPLAY function to summarize the gene numbers that show a significant difference. So we can run the LPLAY function for each cell type. If you look at the DT data, we have 5 lists. Each list has 3 values. Then we can put them together as one data set. Now we can have a look at the data set. You can see we have 5 columns here. The first one is the endothelial cells against the other cell types. The second one is the epithelial cells against the other cells. Then fibroblasts against other cell types. Here is the immune cells against other cell types. And the last one is the parasite smooth muscle cells against the other cell types. So you can see the first row is the down regulation. The second row are the gene numbers that didn't show significant difference for each cell cluster against the rest cell clusters. So the last row is the upregulation. Genes in this row are the genes that we can use as mark genes for different cell clusters. So you can see for the endothelial cells, we have 2042 genes. For the epithelial cells, we have 2654 genes. And here are the numbers for fibroblast, immune cells, and parasite smooth muscle cells. So you can see now we identified the mark genes for each cell cluster. You can get the data I showed you in the table. So next we can produce a heat map to show the top 20 genes for each cell cluster. So you can set the top as 20. Now we can make a list for each cell cluster in order to show the top 20 mark genes for each cell cluster. So now we can order the genes according to the value from the table for each cell cluster. Because we are going to show the upregulated genes, we can set the log FC value bigger than 0, and we can get the top 20 mark genes name from the row names of the DG list. So let's run. Now we can have a look at the top markers list. You can see here we have 5 lists because we have 5 cell clusters. We have 20 top mark genes for each cell cluster. Here are the gene names. So normally we shouldn't have gene names overlap in each cell cluster as mark genes. In case there are gene names overlapped each other in different cell clusters, we can use the unique function to get rid of repeated gene names. Let's run. And we can have a look at the top marks again. You can see we still have 100 genes. That means we don't have gene names overlap each other in different cell clusters. So the top 20 mark genes are specific to each cell clusters. So to make a heat map, we need to get the data. So we are going to use the log count per million for each gene for the whole data set. Let's run. At the moment, you can see it is a large matrix data. We can click and have a look. It contains the log value for all the genes. Next, we can subset the data using the top mark genes. We know we have 100 genes in the top markers. Let's subset it. And you can see now we only have 100 genes. You can see the new data set, we have 100 genes for 30 samples. And we should have 20 genes for each cell cluster. So now we have a data set named as top markers 20. It contains the value log count per million for the top 20 mark genes in each cell cluster. We can use this data frame to produce a heat map. So first we can make an annotate for the heat map, and we can get the cluster name for each cell cluster. We can have a look at the annotation. You can see the columns are the cluster name. Then we can get the sample names as row names. Let's run. We can have a look again. You can see now the row names are the sample names. Next, we can set the cluster color because we have 5 cell clusters here. If we have a look at the annotation colors, you can see it has 5 numbers, 1 to 5. Then we can get the class name for the colors. Let's run again. Now we are ready to create a heat map for the top 20 mark genes in each cell cluster. we are going to use the bitmap package. Let's run this code to create the heater map. You can see we generated the heater map here. We can zoom in to look at the heater map. You can see we have 5 cell clusters, endocental cells in dark color, epithelial cells in red color, fibroblast in green, immune cells in blue, and the light blue color is for parasite smooth muscle cells. Let's make it even bigger. From the dendrogram clustering, you can see fibroblast and smooth muscle cells, parasite, or the cell clusters show similar gene expressions. So the red color show high expression in each cell cluster. So that's top 20 mark genes for each cell cluster. From the heat map, you can see the top 20 mark genes are specific to each cell clusters. Okay, so we finished the video tutorial. And in this video, I showed you how to use Pesudo-bulk RNA sequencing data analysis to identify mark genes for different cell types in human lung. From the data map, you can see the Pesudo-bulk RNA sequencing data analysis produced a reliable result. This is why many researchers perform Pesudo-bulk RNA sequencing data analysis. Okay, I'm going to stop from here. I hope my video can help your data analysis. Please subscribe to my channel if you haven't to do so, and also share my videos with your friends. Thank you, and see you next time.